But I'd like to tell you that everything is just the same as what I told you 2.5 years ago. But you'll see in a moment, that's not quite the state. Some things are the same. I can't help it. I've got to have a little smile in here because as I have come back from the Ukraine terribly opposed to anything modern, their natural streams, their natural production of agriculture, until I challenge where's the money to buy the chemicals. It's natural by default. And 50% of the car population smoke, so natural doesn't buy it with me. This, outside of the Ukraine, what you see on the screen in front of you, I perhaps should hide my head when I tell you, is the response from within the Adventist church. And so I joke a little with what is here. It's unhealthy when you can see all of the other things going. So what I'm going to do is take you through and give you a description of where we are today in the world of biotechnology, plant biotechnology. But I need to drive home a very, very crucial point. You see before you a schematic of a chromosome. A chromosome is made up of two twining strands. Let's say the blue one that you see there is contributed from the male parent. The brownish one from the female parent. Joining those strands are what are termed base pairs, or in better terminology for you to understand, genes. Notice how they are paired. You've got adenine and thymine. The yellow and green color always paired together. There's a cytosine and guanine that are paired together. Notice the colors. This is what drives the function of every living organism. Inheritance from the parents, that then is the code put into the chromosomes and the gene structure. Everything that is alive today. Put in mind the makeup of a gene or these base pairs are small blocks of chemicals. Small blocks of chemicals, which later on I will explain, gives us the opportunity to synthesize, make artificially in the laboratory. So this phobia from within our sacred or brotherhood ranks of you're messing with the essence of life. Certainly we are. Let's talk about that. Oops, wrong direction. Certainly, in reality, even though the right side is a schematic, this is more what a chromosome would look like if you could see it. And variations of some 24 to 48 is typically the range of different creatures that have chromosome numbers. I shared this one with you. What you see here, I shared with you, and I'm going to do this. There's the corn plant, and here is the leaf structure and everything else you can't see without a microscope becoming then a very, very powerful microscope. So today I want to take you, we go from here to the cell of that leaf. Within the cell is this nucleus. Everything I'm going to share with you in the next 45 minutes or so is going to be subnuclear. That is not a ship that goes under the water. That is below the nucleus here. So we have the structure. I think we might have a little loose connection somewhere on this. We'll see what we can do. If I don't turn sideways. That's it. I must look at you. Anyway, up here, folks, we have, and the message right up to the proteins and the amino acids. We then have all of this message contained here in these nucleotides or the base pairs. The shortest in-depth genetics lesson I've ever given. But this is what we're coming to. Where is it? But I want you to keep in mind now as we take a look here, this is probably the strongest message I want you to take away with you today. If I pushed these circles together so they represented reality, you would barely see a color separating them down the side. 3 kingdoms in the creature or the life world. If there's a botanist here, I will concede in the microorganism kingdom there really are 2. But for these purposes, we have 3 kingdoms: plant, animal, microorganisms. And what is the biggest stumbling block to understanding is the gene commonality. The commonality between kingdoms and species. It is so great that many of the devout, purist Eden foods will not accept. Bananas, 80% of the genes are also the identical genes in the human body. Mothers, if you have teenagers, you've got a good reason to say you are going bananas. If you choose. Take it a step further, and I'll take just these 2 examples. Tomato, documented from genomics, 60% of the genes in tomatoes are also the same genes that are found in your body. Same chemical structure. And you'll hear in a moment the functionality that they are arranged in that chromosome. Let's take a moment here. I want to hasten on the picture you see to tell you I have included this not to prescribe the headlines, but to bring to you an understanding of the byline. I put that in the box to the side. This is correct. 98.7% of the genes in a chimpanzee are also the genes in humans. It is that 1.3% of genes that makes the functional difference.
Clive,
Yes.
we're concerned about the gas industry. Would you want to take your jacket off and maybe the jacket rubbing on the transmitter?
That's the best excuse for taking a jacket off I've heard. Thank you. I will be delighted to do that, and if not, we'll turn to the other microphone if you need me to. Sometimes shock value is what it takes. I have an older sister, bless her heart. She is so devout, she has earned her way into heaven by her good works. She and I do not talk about this subject because I'm going to hell. Because I am messing with things that are not true. Not we have, folks. I'm going to change over. If you turn this, if you give me the other one, then I will change over microphones so we don't have an interruption, and we can move to a hopefully a correct recording here. Are you able to turn this one off? I have now. I hope. Okay. This one's on. Thank you. We'll try and do this two-handed. That's easy enough. I want you to understand, within all of living creatures, there is a very strong commonality of genes. That doesn't explain what has been done in biotechnology, but it gives you an appreciation of what happens with genetic modification. Question which is very troubling to the purists, and I tongue-in-cheek, how many dogs did Noah take into the ark? Well, there are those that are very quick to tell me two only. 452 recognized dog breeds today. Look at the diversity. Mankind without a question, since we have been able to select seeds and plant the best looking ones, have also selected animals, have done all of the selection which is considered, if you want, naturally. We'll talk about that a little further. There are 950 plus registered cattle breeds. Belgian draft horses. Where did the little Shetland come from? 18 inches tall. 394 registered horse breeds. Quite acceptable, because they have been crossbred and selected and mutated. I'm driving a point here as we talk about plant biotechnology. So today, I want to take you through, and I'll tell you some of the points here, but I want to drive carefully for you that biotechnology is much like computers. I bought my first computer in 1982, $2,700. I have never paid that price since, and I have never bought a computer since that hasn't been at least 3 times more powerful. And I've bought about 9 or 10 of them. Biotechnology, what was good 2 years ago, is dwarfed right now. I'll give you an example here in a couple of moments. And if we go the other direction here, I want to talk to you about changes and developments. I described for you 2.5 years ago the 3 phases of biotechnology: input traits, output traits, and industrial traits. I want to come back to those. Has biotechnology delivered as promised? Good question. What is the future direction? Then what plants have been modified? I think it's going to surprise most of you when I give you that list in a moment. So we have the biotech phases. What are the differences? Input traits means what goes into a plant that makes it easy to produce. Simple, straightforward. Who then benefits from that? Output traits, quality factors. This is perhaps where you're going to see the biggest hiccup of all. It is what comes out of those traits to benefit, I'm going to use the term that we do in marketing, the housewife, those that shop in the supermarkets. Output traits. Pharmaceutical and industrial. You heard me 2.5 years ago talk about industrial only, of owning up to lumping these two together. Let's then take a look with it. Some examples of input traits to show you why these have been so tremendously successful. What you see in front of you are sliced open corn stalks, damaged by what's called a European corn borer, a little critter on the right that you can see. On average, U.S. farmers plant 80+ million acres of corn every year. This is a major pest problem, because once it burrows inside the plant, there's no way that any chemistry can kill it. So we have then these little critters, these little corn borers that bore also into the ear. They eat voraciously. They leave behind, as a consequence, a lot of frass. That frass, as you might see there, may be hard to pick up in the light, the mycelium, the bluish, whitish cobweb webbing between the kernels. When it turns into the blue color and then into the pink color, it is a mycotoxin, a known carcinogen. $300 to $400 million a year of the national corn crop is discarded because it cannot be used. 3 pounds of that mycotoxin-infested corn
can kill a horse in 15 minutes. Pretty deadly for some species, horses and rabbits particularly. So we have this as an issue. Now, that's a little critter that has bugged American farmers since the days they started planting corn. Notice on the right, a modest infestation to the stem that's cut in half, to the one on the left, which is a corn plant that has been genetically modified with two cells or two genes from a soil bacterium, Bacillus thuringiensis, BT for short, inserted into the corn plant, which then creates an internal insecticide, not detectable by any tests whatsoever. A protein evaluation can tell that that plant has been modified, but it has revolutionized corn production in this country. $1.1 billion worth of extra income to the U.S. farmers today. Mind you, you're going to see a number of figures. Be careful in the context. That's today, because when you see the chart that's gone uphill so steeply, that figure was considerably less 5 years ago. This is one pest. Biotechnology, released for commercial use in 1996. We're in the 12th year of utilizing this. I am going to hasten to tell you, and I'll come back to it, there is not one case from the Center for Disease Control in Atlanta, Georgia, documented, not one case, documented of sickness or death from any biotechnology product on the market today, and almost 80% of the foodstuff that you buy, regular foodstuffs in the supermarket, have some component of a biotechnology plant in them. Some component. Moving on to one more example. The second biggest problem, because it's not quite as widespread, corn rootworm infestations. Corn roots, little critters there that bite and chew and take the roots off. On the right, you'll see the corn plant in high infestation that's been nibbled to almost nothing. What happens when the wind blows? No. What happens first when the rain stops? There's not the root structure to feed the plant. When wind blows, it goes flat. On the left that you see is a biotechnology event that came a few years later, and today, folks, these gene insertions can and are directed site-specific, so that you can only detect that when you evaluate the roots. Yes, this is a biotechnology-enhanced plant. So this is what makes a difference for $1.2 billion today for U.S. farmers in extra income. I will try to address those that want to know, but isn't this a big money-making venture for companies? Aren't you screwing the farmers? Not when you see some of the numbers and what they're buying and what they're getting. Absolutely not right. Let's move on, though. Take a look on the left. No treatment in modest infestation. In the middle is the heaviest dose of the second most potent insecticide that could be put in the soil. There's only one other they use on potatoes. Temic, which is pretty much banned and restricted. This is very potent. Center example. Look at the right. Biotechnology enhanced, where when those little critters bite, they just don't bite anymore. That is what is called an input trait. A couple more. Cotton. You've all heard those songs about boll weevils. Tremendous, as you know, tremendous advantage here that growing cotton in the U.S., I don't have them with me, but in India there are some movie clips I have showing 20 to 22 insecticide applications a growing season. A little less in the U.S., but not that much. Every week, almost, a farmer growing cotton in the southern states and Texas, California, is spraying insecticide on his cotton to produce a crop. That is reduced down to 1 and sometimes 2 by the same methodology of a soil microbe gene inserted into the cotton plant that prevents it from being decimated by the boll weevil. Almost all, except for about 1.5% of the soybeans grown in the US have been modified to resist Roundup herbicide. Almost all of Brazil, and certainly in Argentina. And the funny part of this is they've only been legal for 2 years down there because no restrictions, and they have grown for many years. This is an example of input traits. So input traits, we have got 10 million farmers in 2006 worldwide that grew biotechnology enhanced crops. 90% of those farmers, I'm counting farmers, I'm not counting acres here, are from resource poor countries. You know the headlines. Maybe I'm super sensitive to these headlines, and I read them regularly, that this whole science of biotechnology is ruining the developing world. Follow through here. 90% are benefiting. We had a 13% growth in planted acres of biotechnology crops in the world. And this is arguably a greater benefit to the resource poor farmers than to the American farmers. We'll substantiate that a little more. The demand for biotechnology enhanced crops is growing to the point it almost can't be satisfied. Remember now, I'm still talking about the input traits. Farmers, growers, producers that are producing that fruit or that crop are benefiting. The housewife argues, collectively, the housewife is arguing, it doesn't benefit me. Oh, yes, it does, but not visually. Food costs. When I tell you that in the US, on average, we spend 11% of our disposable income on food. In Europe, that is 42% of disposable income spent on food. This takes in restaurant eating, everything else. There is a fantastic benefit, unseen, unknown, but that doesn't explain some of the points here. I want you to listen carefully for me. 3 minutes. In somebody else's words, he got a pretty good first name, of course, but ISA is an organization that came to life in the early 90s. Oh, let me have him tell you about it. Just listen and watch carefully, please.
Clive James. I'm the chairman and founder of an organization called ISA. It's a not-for-profit organization whose objective is to try and alleviate hunger and poverty in the countries of Asia, Africa, Latin America by sharing technology. 2005 was a very important year. It's the 10th anniversary of the commercialization of crop biotech. We planted our billionth acre
We have the still on input traits, we have the tremendous advantage. You heard some of the figures. You heard some of the drivers, the cost inputs, the conservation aspect, which I'm going to share with you in a moment, that has been fantastic in what this has done. But take a look with me. This, if you picked up, if you heard, Clive James mentioned the fastest adoption of any technology in the farming world, even from tractors, even to mechanization, has been the adoption of biotechnology crops. And largely because of what they do for farmers, what has not been described here. If you think of these internal burrowing caterpillars, unless you can get that window of 48 hours and spray the chemistry on, you don't control them. And they get buried down in the whirl of the eggs are laid down in the whirl of the leaves. So it is almost to a bulletproof situation for farmers from a hit and miss previous situation. Another quick point that I need to add, in South Africa, there have been a number of medical studies that have shown a high incidence, 3 to 5 times higher than the global population of cancer in the population in South Africa. This is because of 2 things related to corn. Beer making from corn, and also the meal used from corn, which in some years is 60 to 70% infested with the mycotoxin mold that you saw in those previous slides. That, no data is available, but expected. South Africa has introduced the growing of biotechnology corn, driven largely by this very, very significant medical problem caused by the staple that the population consumes. That is describing, you can read it as I've spoken, the magnitude of the growth in the input traits. What has it done to the environment? Take a quick look with me as an economist from the UK, unrelated to the biotechnology world.
Company called Economics, which is based in the UK. We have just completed a global review of the economic and the environmental impact of biotech crops as it enters the 10th planting season. We were surprised at the magnitude of the benefits, both economic and environmental, especially the environmental benefits in terms of pesticide reduction and reductions in greenhouse
Again, I remind you, that is the input traits only. Farmers have no question. Success story beyond those of us in the business that would ever imagined. What about the output traits? Those with good long memories will say, you told us 2.5 years ago that's the next wave coming. We're going output traits, enhance the nutrition of products. That brings user benefits. And it's going to be, was going to be, I'm leading you into it now, the second wave. Increased healthful approaches to the foods. Food storage longevity. Tomatoes that'll last 5 times as long on the counter as what they will do now. And retain the flavor. Flavorful products. All reality. All part of what's being worked on. Instead, this phase, you might say, has been skipped. Not entirely, but skipped. I didn't foresee this 2 years ago, but the lack of acceptance of the technology, big one, liabilities. Liabilities, and then economics. Economics is a major issue. Why? Let me give you a moment. First, there are a couple of wonderful examples, and you'll see later on probably 10 or 15 other good examples. I did tell you about golden rice 2.5 years ago, but we'll maybe come back to that. The one I'm very close to is Pioneer, the company that I work for, donated a $5.5 million germplasm base of biotechnology enhanced sorghum. Bill and Melinda Gates put $29 million into an ongoing program in Africa right now. Not enough time to get delving into that, but let's talk about a key factor I have to put in front of you so you have an appreciation of what this takes. From where I sit on budgeting and planning, on average, every new crop that is not new crop, new variety of a crop that is put in the marketplace takes on average $1 million in development costs. That's just part of business. If it's a new corn, new sorghum, new wheat crop, new wheat variety rather, it's about $1 million. Biotech, after that 8 to 14 years for a regular development of a non-genetically modified variety, biotech takes an additional 4 to 6 years. But wait, I've read and heard that this is supposed to cut the time. True, but you have no idea. I just could not describe to you the additional volume of testing, evaluation, and documentation. It is just fantastic. We have to test every product over and over from every known situation, from skin absorption to allergenicity. Is it allergic? Well, you're feeding it to animals. Is it allergic if some child chews it? Everything has to be evaluated. Now's the big figure. It takes, depending on the use and the testing required, today I'm looking at $12 to $16 million for a new biotech crop variety. When you look at modifying carrots, for example, yes, today, carrot seeds are several $100s a pound. But still, the economics make it so that it is a much slower going approach Those of us that get paid to make those dreams or projections for years out figure that in the next 10 to 15 years, the regulatory approach will be much less rigorous. And that will make all of these output traits also having to be tested well. I am not prescribing, I am not advocating reduce testing. It must be in place. But this is the reality of where we sit. And do you know, we've had a couple of corporate irresponsible actions over the last 6 or 7 years. One company put into the marketplace a gazillion bushels of corn that was not approved. It was shown by all tests to have no allergenicity, no untoward effects. It just did not receive approval for human consumption because the studies were not thorough enough for the USDA. But it had been shown very clearly. We had several hundred people after that news item came out that took their box of cornflakes, took their foodstuffs, and they lodged lawsuits over this corporate misstep. Many of them were food items that had no content of corn. The liability, folks, the liability is so great that there is double care on output traits. Is it nutritionally enhanced? And you'll see a few of those in a moment. Is it going to bring any greater advantage? And then to boot, the ability to recoup these investments is so much greater, or the ability is so much reduced, so much greater in difficulty. Output traits are coming, but much more slowly. And I think I've spoken ahead of what I'm going to tell you, the summary. It's stalled. Bypassed almost, but that's not correct, really. It is stalled and down this siding with work continuing, but with not the same fervor that was 3 years ago. Technology acceptance is lacking. In many quarters, you've heard the very positive of the input traits, but research is continuing more modestly. Wave 3, the third phase, is the one that is fired up and steaming down the tracks so fast today. You don't hear of it, you are not aware of it as much, because who knows about plastic? Who knows about steel substitutes? All of those things in the works. Not nearly as fast. So you will have to wait a few years longer for those nutritionally enhanced products. But I do stand here and tell you that where I sit, we have just released for commercial use several different soybean varieties with the oil enhanced to have the trans fats removed, the amigo fatty acids added. They are coming, but you're not going to see it as a wave. You'll see them more as a trickle. You'll see it first in these more extracted items. Those are coming. They are being produced as we speak and will be on the shelves in the next 18 months or so, that you will be able to look at those labels. Guess what? Are you one of 7%? Only 7% of the population reads labels of the foodstuffs they buy. And I often wonder if those 6.5% are from the Adventist world, but they're only 7% on survey results. So we look, the cost is outweighing the potential advantage. Yes, there is nothing wrong with making a profit. There is all things wrong with making obscene profits. But we'll come back to, if anyone is interested, at question time about what does it cost farmers for their input traits, and I'll talk to you about that. It is not obscene. They are making 3 times more than what it's costing them to take the technology. Biotech Phase 3. This is where pharmaceuticals and industrials, and we've divided them. I've spoken about the dividing of these because they lump together. This is where, do you know today, that 31 or 32% of your medications are they are formulated from biotechnology enhanced bacteria that are put into a tub and fermented? Purity is the difficulty, consistency, and it takes a lot of refining. The same proteins developed within plants have the purity issue taken away, or the lack of purity taken away. The ability to be more consistent is very much part of the plant pharmaceutical production, or the proteins to produce those pharmaceuticals. And the major concern we have is a company 2-3 years ago in the state of Nebraska planted these 2 fields of, under special permit, corn plants with enhanced protein production for pharmaceutical development. 12 months later, after that crop was harvested, an inspector happened to drive by one of these fields and noted in his book, now growing soybeans, but in that soybean crop, if any of you know farming, there were these volunteer corn plants. The worst situation that could have occurred. That cost multi-million, I think $110 million was the settlement from that for all the soybeans and contamination. This is what cannot happen. Today, there is no pharmaceutical producing plants grown in open air environments. The move has been made in southern Indiana. There are many, many empty salt mines. Greenhouse environments have been set up there, only in greenhouse, but there is a concern about pollen escaping from the greenhouses. So very, very major concern on liabilities for if you had a protein escape that was a large component of some medication that had an allergic reaction, you wouldn't be happy if you weren't alive to sue for it. And that's the significance. So industrial is moving at such a pace, I even have trouble keeping up at times. Plastics, polymers. This world is, nothing moves without a polymer. In every phase of everything, from auto mechanics to polymers, are a component of so many things. One example on our time limitations here. Taking a gene of all things from a silkworm, putting it into a tobacco plant. Why tobacco? It is one of the most easily transformed, in other words, genetically modified plants there is. There is a component that can be made with the fibers extracted, the proteins, that there is a fiber-like component 5 times lighter than steel and 8 times stronger. The U.S. Army is currently testing this product, of all things you could imagine, flak jackets. So there are things coming that you could only imagine in your dreams. Industrial is where we have so many things on the horizon that the renewable aspect of being able to take over. Today, another example I'll talk about, I think it's coming up here, ethanol production and taking over from the oil consumption, so many things. I've touched on all of these points of farmer production. Interestingly enough, it takes a very small quantity of plants to produce the proteins that are used in medications. Very small quantity. The prediction is that less than 1,000 acres, under the ideal conditions, would supply all of the proteins for the pharmaceuticals produced in the United States at the moment. That was one of the projections. Not all projections are right, but it gives you an idea of the smaller scope of that. So we've got a success story on the input traits. We have got a developing story, success to the greatest on the industrial traits. And all of the others, I think, are going to come along quite nicely, but at a much slower pace. Let's take a look. Here are some of the things that are already in place, already developed, but unfortunately, they likely are not going to be released. Because if you sent Junior out to pull a banana off a tree, we spent some time in the tropics, to get his hepatitis vaccination for the day or the week, and he ate 3 of them, that has the problem situation, overdosing of some medications. But we do have bananas with vaccination doses, biotechnology enhanced bananas. I put this in because it's now transferred to plants, but we have cows that are producing proteins at 3 to 5 times the quantity of normal production that brings alleviation of cystic fibrosis symptoms. It has also been transferred into plants. Anti-malarial vaccines in bananas. Oral diarrhea. Do you know there are some 3 million children a year die from just plain diarrhea without being treated in the developing world? Tremendous losses. Rice with an allergy vaccine. Tomatoes we've skipped by with SARS vaccine. These are already in place, developed, available to be used, but it is the facilitation of them that has too many problems for this to happen. Corn, now this is a different one. This will be put into production for pigs, for pig production. I come from the state of Iowa, number 1 in corn production in the nation and number 1 in hog production in the nation. And it has a very significant application. Farmers will mix their feeds, and they can mix this corn at a ratio that gives that prevention of diarrhea in these little piglets, and so on and so on. This is pharmaceuticals, folks. Now let's take to the next view here that we need to look at on unique advances. Unique advances gone through input, output, industrial, and pharmaceutical. What is happening? We have a situation, and you're going to ask, and that's fine, I welcome it. Can you patent genes? Well, if you don't think you can, go have a look at the patent registry. It's absolutely happening, and more being able to work with freedom is locked up by patent holders that have patented either a process, a gene, or some part of the technology. Fees to negotiate are difficult. Well, the negotiation for the use is difficult. The fees are high. And it is, I come from a company where in 1988 we had 0 collaborations. Today we have a computer database with over 12,000 collaborations, licenses, dealings with other companies, other entities. And you talk about trying to work through a maze. We have groups of people who are dedicated to doing this. It's increasingly difficult to innovate because not one entity has got the lock and the key. And so it happens that a frustration of where you go. We have technology developed, but we don't have permission to put it in with a certain process. And all of these interactions are locked down by somebody who thought that they could make some money somewhere. So what I want to talk to you about is what is happening. What is happening? This is one that I happen to be in the middle of, which is exciting. New technologies. You're going to hear me say several more times. There are walls being built, and there are going to be tunnels dug underneath those walls. Gene shuffling, molecular markers, probably never heard of it. So let me take you very quickly for a walk through. You see here, you've got the chromosomes again and the genes between. So then look at these schematics of segmenting this chromosome here so that we've got and understand now, not just one of these makes a gene. There will be a collection of 2 or 3 or 4 or 5 or 20 in this case. So you're looking at a situation where we're going to shuffle these genes. It's a new technology. 2, 3 years ago, under experimental testing, and this is a good success story of what I call one of those tunnels under the wall that has been built by so many blocks in the legal arena. So we have this DNA. We then take them, and in a test tube, as you see those little stars, we break them apart. It's a process with chemicals and a little heat. They get broken apart. And it's much more laborious to tell you how this happens, but broken apart into gene fragments. Then those gene fragments are broken apart. This all happens on stuff that you never could see with your naked eye. They are broken apart as you see the schematic of there. Then they cool down and they come back together with holes that are developed. And this is where they then fill in those holes naturally. And the functions of those genes are completely different, internal to the plant, of their own genes. So we have a regulatory situation with any genetic modification. Here we are going by one wall. Next wall is on a technology that one entity might be blocked by another from doing anything with, to a new way of gaining very successfully a result. And this gene shuffling is where the genes will produce different proteins. Those different proteins then have a different result. I would like to pause and tell you the details and how, but you're going to have to come back if you have an interest with a question. It happens. It is happening right now. It is so successful. We're able to select these shuffled genes in such a way that you'll see the ones that are not successful there, they are put aside, and then the successful ones that show some response, as you see here, circled in green, are selected for a second round and a third round. The same process. Look with me. What happens? Taking an ineffective gene, and I'm giving you a schematic here of a real situation. An ineffective bacterium, soil bacterium gene, and reshuffling them 11 times. Look at the actual data. This is the actual data of the parent genes right on the left. You can't even see the efficacy here. Doesn't even register on the scale of efficacy of any production reaction. Shuffle them 11 times, and that has been increased in efficacy 8,000 times over a short period of only weeks. And then, instead of years and years of doing, the shuffled gives us response very quickly. In this case, this ineffective soil bacterium that was locked down by patents and insertion technology restrictions has now been able to be shuffled in a different manner completely to give, in this case, where a spraying of chemistry, and this is not just one chemical. There are 57 different chemistries. This here will kill every time, and then look what happens on the same plants that have had their genes shuffled. A segment, small segment of their genes shuffled. I'm giving you an example of technology that to most is going to seem so far out, it's almost unbelievable. But it is a technology that then is giving a similar result, but the magnitude is far greater than anything that's happened so far. I happen to be right in the middle of this now, and there's the corn plant with the same chemistry applied, and anyone that knows those chemistries and rates, and that is only a fraction of what the resistance conferred. Again, this is the bicycle of biotechnology with this type of intervention or change in what to do in making these changes. So many other things will come from this, so that you hear these wild stories you think they are of producing oil substitutes. Don't doubt them too much, because hidden behind all of this work, there is so much more already done you probably won't see for the next 15 years. But it is work going on like this that is bringing so much advantage to this business of environmental containment, cost savings, and what we as humans need to be doing for the environment in the world that we live in. One other last point here. This is an intriguing one, molecular markers. It is a non-biotechnology approach that's giving us the same results. As you look at, in this side on the right, you'll see in this picture fusarium-infested corn plants that the first puff of wind blows over. Same plants grown 3 feet away, developed through a molecular marker that has selected the trait, the gene in that plant that controls, or the intensity of protection is increased to the point that it withstands that infestation. This is, I'm giving you a smattering, just a slight smattering of technology as we go. Patenting nature. I'm jumping a little now. I have finished, shall we say, an update of the technology. I'm going to try and anticipate, but you can't patent those things. The Lord made all of nature. You can't touch it. The first question you need to ask yourself, what is patenting? What is patenting? Look with me. Patenting does nothing but confer restricted use. It does not give ownership. And the patent laws of this country and globally in the Western world pretty much are either 17 or 19 years. 17 is the most common. Chemical company says, raises their hand, says, we've got a new chemistry under development, we are patenting it now. And this has to happen at discovery, because if that discovery leaks out, 3, 4, 5 years down the road, somebody else can pick it up, and you've lost everything. And then that is typically 5, 6, or 8 years before the product is ready to be commercialized. So the patenting does not give ownership. It gives protection. It is an economic protection. Every day, every month, I'm dealing with products that come off patent. You and anyone else can take them and develop them for yourself. These are chemistry products. This is biotechnology products. So the patenting, as much as it goes against the grain of everyone, you can't patent nature. It is an economic protection necessary. In the seed world, which I'm very much part of, we patent our varieties, our hybrids, to the point that you, Mr. Jones, cannot go and crossbreed that. But you can in another 10 years when that patent runs out. But by then, the newer products are much more superior. I've covered that in the points here that the patents do expire. And so patenting, while it locks things up, is so absolutely necessary when we're dealing with the extent of products that we are. I looked at one of our checklists just this past week or a couple of days ago, where I sit. If I add up corn at 18 products under development, soybeans at 17, there are several others we've got going, of about a total of 34 biotechnology events under development times $15 million. It is a situation that you can't do if you don't have some protection. You've read about people that have broken patents and gone. The man that invented the intermittent wiper collected $200 million out of Ford and GM after they had stolen his patent. If you read those news, it goes on in the business world. That's what patenting's for. And so it is for recouping investments. I make no apologies for patenting, but I do want you to understand the explanation. Future. I need to move fairly fast here because I'm going to wrap up on this here and tell you we have the answer to most of the activists' opposition to plant biotechnology. The pollen is going to cross-pollinate. Look on the screen. Terminator gene, as it's been dubbed. That is not the correct name, but I use it because everyone knows it by that. There is the ability, it has been in place for 6 or 7 years now, where plants are modified, they are fertile for 1 generation only. Then they will not produce a grain crop after that. That is unacceptable ethically to everyone in the world, and those where I sit say, we will acknowledge that because as the African subsistence farmer does not use the types of crops that we're developing today, they use stuff that was around 50 years ago, except you did see in the video, that's changing. Ethically, it is a non-starter. So, my tunnel, again, under the brick wall, look with me. Gene switch. The activists have got to squirm every time something happens like this because you could say the Terminator technology of shutting down reproduction in a plant after it produces the initial grain crop or seed crop. Now, we have the technology built in, available, that will turn the reproductive situation off in a plant after that first crop, with one caveat. If you buy this chemical, you can spray across the top of that crop and it will reproduce for 1 more year. Then you can buy it the next year and reproduce for the next year, which overcomes the major opposition of shutting a crop down completely. It will still produce a seed crop. It will still produce a wheat harvest. It will have those grain, but that grain will not contain the gene for the next crop that protects it against an insect. So if you want it, you then have to do this technology. That's just one of the things happening. Gene shuffling, I've taken you through that. I want to then just take you, it amplifies that very ineffective gene and to the mini cassette or the mini chromosomes. Would you believe where we take one gene from any various organism, now we put them together in a cassette that will give 2, 3, 4 different effects. And the big side advantage, it does not disrupt the current genome within that plant. It will give enhanced ability of nutrition. It will give enhanced insecticidal properties for insect protection. Anything that can be dreamed of for producing that plant to a greater level, greater root strength, brighter red color of the cabbage if you want it. Anything that's possible, it's coming, folks. I'm doing a very brief situation here talking about these multiple stacks that can go together. So what I would like to do as we finish this session, let me just give you the one last summary here on what the future is. Opposition is not going away anytime soon. My sister has many like-minded people out there. Wonderful person. Just a little misguided, I think, in her theology, but that's okay. She's a devout Christian, Adventist Christian. But it's not going to go away. And we might have time to talk about all the technologies in this world we as humans have resisted tremendously in our resistance to the motor car, to electricity, to vaccines, to all of those things. Technology will circumvent any contention. It will get around it. We have so many things that are coming around it, underneath it right now. You heard me briefly on the patenting, and it will enhance the environment. Go to India, and what do you see in India? Of all things, you would think they had all the plastic in the world. India has plastic bags blowing in every direction in every part of that country. Just think if after 6 months every one of those dissolved. That's coming, or it's here now. It's just not widespread enough. Proteins for medications. They will move. There is no doubt for the advantage that brings. Proteins for medication development and production will move to plant-based supply. Food enhancements are coming, but they're going to be much slower. And that, folks, is where we are with industrial components being a tremendous boon to our environmental concerns. What is happening from all of the oil-based or the petroleum-based products we have are now being replaced by plants enhanced to give the same polymer construction for the production of those things. And it will increase and it will flourish, and I think it's a situation that is not going to go back anywhere. It is going to go forward, and I want to just wrap this up. If we make this the end of the audio presentation, I just, for the amusement of it, you'll notice that I chose a UK economist. The UK, Britain, does not allow biotechnology plants in Britain. And these folks are studying, and you saw his report. Clive James is a Belgian by birth, but he's living in Canada now. I want to have you just close this session down for the audio recording. We'll finish at this point. I'm going to play you another 3-minute clip of a British politician. Intriguing, fascinating, because he has nothing to do with biotechnology. So if you will allow me, I understand, Jim, we're going to break now, have 10 or 15 minutes. If you come back, and Jim's going to tell you the time for coming back, I want to start off the question period by showing you 6 or 7 minutes of a list of biotechnology-enhanced plants. It'll go through a whole lot of pictures and tell you what it's about. I'll start it with that, but first of all, just bear with me as we hear from Lord Taverne. Okay?
So I think environmentalism is a very, very important cause. But it's got to be based on evidence. And I got more and more interested in the whole question of genetic modification, because that, in a sense, has been the central battlefield in which the anti-science and science forces have locked horns. The more I've looked at genetic modification, the more I've become an enthusiast for technology. The evidence is fairly clear on certain points. First of all, there can be no doubt that so far there is no evidence of any damage to health, any danger to health. This is a point which the opponents still haven't grasped and won't admit. The third world academies, the Indian, the Mexican, the Brazilian, the Chinese national academies, apart from those in the United States and Britain, they say that there's no evidence of any harm to health or so far to the environment. It's reduced, quite clear, it's reduced the use of pesticides. It produces greater productivity. And if it reduces the amount of farmland you have to use, it can actually be very beneficial to biodiversity. It's very beneficial to the environment. And indeed, there are any number of developments which could make a huge difference to the fight against disease in the world. And here are these people who are fighting against it, who claim that they're benefiting the third world. Hypocrites they are. I mean, it is extraordinary how they don't even look at the evidence. They're just dogmatically opposed to the evidence. Some people take the view that you must always distrust scientific research produced by companies. My answer to that is good research is good research, and you can have very good research produced by companies and very bad research produced by the public sector, and the other way around. What matters is, does the research that's being done stand up to testing? Has it been peer reviewed? Is it reproducible? Those are the tests. And if you look at the crops, BT, cotton,
That has just triggered one last comment, if you'll allow me. The U.S. is at the forefront of biotechnology enhancement of plants. His last comment, I have to be guarded where I make this comment, but it is a direct reflection, of course, of the American innovation, but the educational level of the population. There is no other country in the world that has that 64% of the population that has attended college or university at some time. And it is that greater acceptance, as well as combined with, relative now, the very high level of acceptance and trust in the government regulatory processes in this country compared to the European countries where it is not transparent. And so it is very much this country that has been at the forefront of the technology. Thank you.
Are we ready for the break then? You want to do the break then? Okay. Have you enjoyed what you heard?